Hello, this is a quick run through of IB Chemistry, Option B, Biochemistry for higher level, in anticipation of the examinations this week. Lastminute.com, let's go. Michaelis-Menten, or Michaelis constant Km, is equal to half its value at half Vmax. So Vmax is the maximum point where all the active sites are bound, though the enzyme is saturated. There are no more active sites left. With half of this value, notice its rate of reaction on the y-axis and substrate concentration on the x. Those are often mixed up by students. If it asks you to work out Km, it is half of Vmax. Vmax is on the y-axis. Say it again. Vmax is on the y-axis. Take half of that, not of the substrate concentration, to get the Km. Use the graph to determine Vmax and Michaelis-Menten constant. 0.8 is millimoles per decimeter cube per minute. Half of 0.8 is 0.4. Then we drop the arrow down and find it's 0.7 millimoles per decimeter cubed in substrate concentration when half of the active sites have been bonded with. Vmax tells us how fast it's going to catalyze the reaction. Don't forget the catalyst is returned unchanged at the end of the reaction. Low Vmax, it doesn't really convert very much, and a high Vmax means it's going to convert a lot of substrate into whatever the product happens to be. Enzymes will vary depending on their affinity for the active sites or the substrates. A high Km means a low affinity, a high Km means a low affinity, a low Km means a high affinity for the substrate by the enzyme. Inhibition, competitive and non-competitive. Inhibition depends on the active site, the allosteric site. Competitive bond at the active site. They're competing with the substrates. They have the same architecture of the molecule, and therefore they can bond into that lock that the key is going into. Once they bind, they do not form products, they just block the site, so no more substrates can bond. You can see the bond model at the bottom of the screen. How do you combat competitive inhibitors? You increase the concentration of the substrate, so you flood the enzyme with substrate, and then that will lower the inhibition and therefore increase the efficacy of that particular enzyme. How does this affect Km and Vmax? Well, Vmax is not changed. Km is increased as it takes a higher substrate concentration to reach Vmax. Let's do that again. Vmax is not changed, as there is still a substrate concentration where full enzyme activity can be achieved. So with the competitive inhibitor, we are going to what? We're going to slow the rates. The rate is going to increase at less rate but reach the same maximum point. That means Km is increased. Km is further along on the x-axis. Competitive inhibition. Non-competitive, this one's going to bind at the allosteric site on the opposite side and deform the enzyme. The effect now is clearly quite different. Here it was slower to get to the Vmax, therefore Km increased, and now it doesn't actually reach the same because the non-competitive has actually deformed the enzyme, so nothing can actually bond with it. Vmax is decreased, but Km is unchanged. Vmax is decreased, but Km is unchanged for non-competitive inhibition. I would put money on that question being asked this year. Look at that table, learn it, deal with it, move on with your lives. Compare and contrast competitive and non-competitive inhibition. Great IB worded question. Not very buff. Buffer solutions resist changes when small amounts of acid and base are added. Here we have ethanoic acid with sodium hydroxide. Sodium hydroxide is a strong alkali. Ethanoic acid is a weak acid. We know strong is going to completely dissociate and weak is going to partially dissociate. However, when they react together, the sodium will react with the conjugate base of the ethanoic acid. The sodium will react with the conjugate base of the ethanoic acid, forming sodium ethanoate. Once a small amount of the conjugate base has been removed, equilibrium will re-establish more conjugate base, so this will go to completion. Notice it's not an equilibrium arrow. We know at the beginning that we have 0.5 moles, 1 mole of ethanoic acid and half a mole of sodium hydroxide. At the beginning, ice table, we can't have any of the sodium ethanoate or any of the water, so they're 0. If we find there's 0.5 moles of ethanoic acid left, then we must have used all of the sodium hydroxide. Conversely, if we only had 0.5 moles of sodium hydroxide, we can only use 0.5 moles of the ethanoic acid. That means we can only make 0.5 of the sodium ethanoate and of the water. The final solution has equal amounts, therefore, of weak acid and the salts of the weak acid and the strong base. This is a buffer solution. We have excess ethanoic acid, excess, because we want more of the conjugate base when it's been used up, and the sodium hydroxide is limiting. The likes of this on paper 1, which one would form a buffer solution. How do you make an acidic buffer? Well, I've kind of just explained it. Here's our sodium ethanoate that will completely dissociate into Na plus and the conjugate base CH3COO-. In an excess of ethanoic acid, making conjugate base plus a proton. So if I add a proton, it will react with the conjugate base and make the ethanoic acid. Well, that's not strong, it's weak, so it's going to have a higher pH than sort of 3, 4, somewhere around there. If I add base, what's it going to do? The base will abstract the proton from the ethanoic acid. It'll make more of the conjugate base, which is pretty neutral, and water, which is always neutral. OH- will also react with a H plus and make water, which is, as always, neutral. A basic buffer, conversely, it is the same theory. We need a weak base. Weak base is here, ammonia. Will abstract a proton from H2O, giving you NH4 plus and OH minus, but that's an equilibrium. It's a weak base, so equilibrium will very much rest to the left-hand side. Mix that with the salt of the base, in this case ammonium chloride, complete dissociation. So if I add H plus, I'll make more NH4 plus ammonium. And if I add OH minus, I'll make more NH3. Weak base does not change the pH any great deal. And water, neutral, as always. Here's the good old Henderson-Hasselbalch. pH is pKa plus log salt over acid or minus acid over salt. They sometimes give you the pH. Sometimes give you the concentrations to work out pKa. Sometimes give you the pKa and the concentrations to work out pH. Sometimes give you pH and pKa to work out the ratios of conjugate base to the acid. We assume that it's only partially dissociated, and we assume that the initial concentration is the equilibrium concentration of the weak acid. Could be a 1 marker at the end of your calculation. What are the assumptions implicit in this calculation? The initial concentration is equal to the equilibrium. We've said that. Flying through here. Calculate the mass of sodium ethanoate required to make 1 mole per decimeter cubed of pH 5, starting when 0.1 of ethanoic acid and solid sodium ethanoate. It gives you the pKa, so we want the mass. So this is the concentration, or the amount, of the conjugate base. So, what's the mass? Well, this is a Henderson-Hasselbalch. Plug the numbers in. We've got the pH, which is 5. pKa was 4.75. A minus, we want to know, that's the conjugate, that's the sodium ethanoate, over 0.1, which is given for the concentration of the ethanoic acid. Plug the numbers into your calculator. You find it 0.178 moles per decimeter cubed in 0.1, times it by the molecular mass of the sodium ethanoate, giving you 14.6 grams. A typical use of the Henderson-Hasselbalch equation. Don't forget the assumptions. Move on. 2. Calculate the pH for 50 cm cubed of 0.2 mol per decimeter cubed ethanoic acid and 50 cm cubed of 0.1 mol per decimeter cubed sodium hydroxide, pKa 4.75. Pause the video. Go. 1:1 reacting ratio. The moles of sodium hydroxide 5 by 10 to the minus 3. The moles of ethanoic acid 1 by 10 to the minus 2. The moles is volume is divided by 1,000 times m. So the final concentrations, you've got 5 and 1, so you've taken away 5. So very similar to the one at the beginning. So you made 5 by 10 to the minus 3 of the sodium ethanoates. So therefore the sodium hydroxide is the limiting reagent. Plug the numbers in. Henderson-Hasselbalch. pH is 4.75. The mistake in the solution, it should be 0.05, not 0.5 in the displayed calculation. Aha. A few more cheeky examples. pH of the aqueous solution contains 0.8 moles per decimeter cubed vita ionic and 0.2 mol per decimeter cubed anionic forms of serine. Pause the video, have a go. 0.2 anion, 0.8 conjugate base, plug it in, 8.5. Example 4, you can read it. Pause the video. pKa is minus log to the base 10 of the 1.8 by 10 to the minus 5, so it's 4.74. The concentration of acid is 0.09, anion is 0.11. Plug the numbers in, salt over acid. Final answer, 4.83. Pause it if you want to review that. We're moving on. If I add something to the buffer, if I add 0.01 moles of sodium hydroxide into the buffer in the previous example, into here, what's going to happen? We had 0.09, so we take away 0.01, 0.08, 0.12, because if we add sodium hydroxide, it will completely dissociate, and it's a 1-to-1 reacting ratio. Plug your numbers in, the answer is 4.92. Again, pause, and moving on. Beer-Lambert law, log to the base 10 IO, instant rate over I will be E, the molar extinction coefficients, times the path length, times the concentration. Intensity of light before and after, I've mentioned the other three when I was talking. Beer-Lambert law, we use a calibration curve. You can use this with a colorimeter. You might use this in your IA. We know that absorbance at a specific wavelength, in this case it's 635 nanometers, and we can plot where the concentrations give us those absorbances. The greater the concentration, the greater the absorbance for most solutions that are coloured. We can then take an unknown, we can extrapolate from the absorbance and work out the concentration, as you can see here. This is because absorbance is directly proportional to concentration. There's a video. Protein concentration is done by UV visible spectroscopy. That's just a factoid, you need to know that. UV vis for assay of protein concentration. Here you can see a typical spectrometer readout. Some of you did do this for your internal assessments. Chlorophyll appears green because it absorbs at 430 and 660 and reflects the remaining wavelengths. That's why it appears green, because it's what it absorbs. What's left is what is transmitted. We're going to use a spectrophotometer, source, collimator, monochromator, wavelength selector, sample solution detector, and readouts. Maximum wavelength, or lambda max, is the greatest absorption of light at a particular wavelength. This time it's 626. And we can use the calibration curve to get the unknown protein concentration using Beer-Lambert law. Nucleic acids: RNA, ribose, DNA, deoxyribose. Okay, RNA single strand, DNA double strand. Compare and contrast RNA and DNA is another predictable question. They're both polynucleotides. They're composed of nucleotides with a sugar phosphate backbone. Double helix for DNA, sugar phosphate backbone, 3 times now. RNA is single-stranded, and the 2 strands are actually by hydrogen bonds between nitrogenous bases for DNA. Here's a typical subsection of the DNA strand. We have the nitrogenous base. You can see nitrogen in the heterocyclic ring with the 6-membered ring and the 5-membered ring connected to a sugar and the phosphate group. Look at the pentose sugar. 1 is from the right hand side of the oxygen in the heterocycle. 5, 1, 2, 3, 4, 5. Go down the bottom of the ring. 1, 2, 3, 4, 5. They are bonding together because they've got a very reactive phosphate group with a condensation releasing a molecule of water. C1 to C5 is the bonding in this particular nucleotide. Between the nucleotides just mentioned, it's a condensation reaction. It could be called a phosphodiester linkage. Why is it phosphodiester? Because it's phosphorus oxygen and phosphorus to oxygen to carbon on the sugar molecule. Phosphodiester linkages, condensing here, as we just mentioned. Don't forget water as the product. You can see where the H and the OH come from. Does it matter which OH and which H you use? No, of course not. Just don't forget the water. Here are the bases. They're in your data booklet. You don't need to learn them. There they are. Nitrogenous and purines down here. You can see that there's 1 ring and 2 rings for guanine and adenine. DNA, we have cytosine and thymine, but RNA, uracil and thymine. If you don't know that yet, please just remember it for Friday. Base pairing, we say AT and GC. AT is 2, T is 2, GC is 3, GC is 3. Guanine, cytosine 3, adenine, thymine 2. Hydrogen bonds. Okay. DNA is pentose, DNA is deoxyribose, and RNA is pentose sugar. Deoxyribose does have an oxygen, carbon 2. You can see that clearly upstairs on the diagram. Here we have a lovely diagram of our single and double helix. Moving on, a video if you want to watch it. Protein synthesis is done by transcription. Proteins are passed from DNA to mRNA, and it's done by RNA polymerase. RNA polymerase is the enzyme in protein synthesis. Thank you, biologists. You named them after their job, their function. One is initiation. Initiations separate the 2 DNA strands, so they're snipped apart from the parental DNA, making 2 strands of DNA. Replication base pairs, always TA and GC. And then transcription with the RNA polymerase here. We have the RNA strand coming in, and the matching nucleotide is added from the new strand coming in. DNA is rewound, and the new strand of DNA is made. If I were an enzyme, it would be DNA helicase. I'm not going to finish that. The newly formed mRNA leaves a nucleus for the ribosome where translation takes place, decoded by a ribosome to produce a polypeptide chain. And here you can see the constituent amino acids going into the polypeptide chain here, being synthesized with mRNA. Translation of the triplet code, there are 64 permutations. You need to know that. Yes, you do. And the 64 permutations come from the 20 naturally occurring amino acids. Those numbers need to be known. Complementary pairs, I've already done this. RNA has U, DNA does not. DNA structure, TAGC, hydrophilic backbone, and non-polar hydrophobic bases with their planar structures stacked inside the double helix. They are flat molecules in the center of that chain there. DNA is very stable because of the base stacking. You can see the bonds that were going between the GT and the AC, and the natural DNA and the planar London dispersion forces. Great question. AT and the GC. There we go, just said that. Base stacking, cellular environment is aqueous, therefore polar, but the non-polar bases are charged when the phosphate maximizes the solubility of DNA in your systems. Go DNA. Tightly packed in the nucleus of every cell. Special proteins called histones, and these nucleosomes coil and stack together to form chromatin, in which they form larger loops of coils to form chromosomes. Packaging, histones, chromosomes, chromatin, chromosomes. You need to know those words. If you don't know them yet, just remember them now and forget them on Saturday. Don't need to know that. Don't need to know that. Very good. Biological pigments. Colour compounds, because they have conjugated ring systems. We've already seen this readouts, this spectrum from a spectrophotometer in an earlier slide. Enormous molecules here. We have loads of pi electron density throughout, and we're looking for the alternating single and double bonds to give us the phenomenon of seeing colour in the visible region of the spectrum as human beings. Okay, don't forget the colour wheel. You've all done that in higher level periodicity. Moving on. The more conjugated is, the longer the wavelength absorbed. What do I mean more conjugated? More extensive conjugation in the chain and in the rings which are attached. So the top one, beta carotene, absorbs blue, therefore it transmits orange. Retinol absorbs violet and transmits yellow because it's shorter. Okay, so longer wavelength is absorbed, the more conjugated the pigment is. The larger it is, the longer the wavelength. We just said that. Carotenoids come from plants. Here we have beta carotene and lutein. We can see here clearly lots of alternating single and double bonds and loads of pi electron density. You all remember what pi electron density is, right? Will it be fat or water soluble? Well, for water soluble, we're looking for polar, and for fat soluble, non-polar portions of the molecules. Very good. They are accessory pigments. It's extended by such as carotenoids and cannot transfer sunlight directly, but instead pass their absorbed energy to chlorophyll, which is great because the plant can grow. Missing pigment, that's albinism. Here we see a white koala, very Australian specific, and also in human beings. Oxidation of carotenoids. Oxidation just means that you're going to add oxygen. You need the presence of oxygen for it to happen, and normally photolysis, so it's photo-oxidation. You can stop it oxidizing by preventing its exposure to air, so pack your carrots in CO2. Anthocyanins. Anthocyanin with a sugar. Handle this glucose on here. These are water soluble. You can predict that because there's loads of OH groups and oxygen going around. They are called flavonoids, always highly conjugated systems with lots of delocalized pi electron density. Are they soluble in water? Well, the polar hydroxyl groups would suggest that they are. Yes, indeed. Here we can have them as pH indicators. Cyanidin has less conjugation in low pH, so it's red, and more conjugation at high pH, so it's blue, and it's a beautiful violet in the neutral range of the pH scale. Anthocyanins are stable. They form deeply coloured coordination compounds with iron 3+, aluminium 3+. Heated up, they do fall apart pretty quickly. Chlorophyll and heme are in your data booklet. Porphyrin are chelates. Chelates, remember chelates, it means that they have numerous centres with which they can bond with lone pairs of electrons on neighbouring atoms, so it's vacant orbitals on the transition metals. Excuse me. And you remember all about ligands, I am sure. You don't remember ligands? Time to look it up. Prosthetic group of heme is the non-protein part of the protein molecule, and got iron 2 to iron 3 shuttling backwards and forwards, electron transport, so you can move oxygen around your bodies. Hemoglobin, this is the quaternary structure, and it's bonded to iron 2 at its centre, transporting oxygen, as we just said. Hemoglobin, ha ha ha. Here's the structure, myoglobin. Will you ever have to draw that? No. Is it in your data booklet? I think it is. If it's not, they'll give you it. Never going to ask you to do that. One heme group is in the muscle cells, and it stores your oxygen. So here's the summary of porphyrin compounds. Pause the video. Test yourself. Learn it. Move on with your lives. Cytochromes, they contain heme. This is this long, extended chain of 1, 2, 3, 4, 5-membered rings, and then iron coordinated at the centre. Cytochromes become successively reduced and reoxidized as they pass electrons. Electron transport change, you lovely biologists out there. Oxygen will saturate hemoglobin. If you get carbon monoxide there, it will bond irrevocably with the hemoglobin and not allow oxygen to be transported, and then you pass away. Oxygen's hemoglobin is cooperative. The first oxygen binds with low affinity but increases the binding affinity of further oxygen molecules. That's in bold for a reason. I remember doing this in the lesson. The first one bonds, lowers the affinity, but increases the binding affinity, so more oxygen. So even if you got a little bit, it will allow it to get you some more. So it's a good, excellent adaptation of the human body to get more oxygen in quickly. Okay, here's the dissociation curve, saturation of hemoglobin, and different partial pressures. So the more that there is, the more saturated it gets, up to a maximum. Okay, it depends on whether you're young or old. It depends in which part of the body you are looking, whether tissues or lungs. And then here we have the effects of temperature. At a higher temperature, it's going to go at a quicker rate. Nothing changed there. It always has done, always will do. pH, at a lower pH, you get more CO2. Why? Because CO2 is quite acidic itself. It's forming carbonic acid. And then, of course, babies' fetal hemoglobin goes much more quickly and get much more in at lower pressures or lower concentrations. That means the fetal blood and placenta can take up oxygen from the mother's blood. Do you need to know that? Yes, you do. Explain why fetal hemoglobin has a greater affinity. Well, 2 marks. Receives oxygen, less sensitive to inhibitors, and differs in amino acid sequence. How did we know that? Because we did our homework. That's why. Carbon monoxide, hemoglobin. I've already mentioned that one. And you can see carbon monoxide just completely saturates it. That's it. Oxygen does not get a look in. Okay. Let it go. Let it go. Colour stability of heme. Purplish red of meat is caused by myoglobin. You see it in the shops stained blue to red as if it's never been an animal in the first place. And you can see here, which one would you buy? Or would you buy it at all? It's all about +2 and +3 oxidation states, but maybe that's not what you're thinking when you're thinking about your steak and chips. Stereochemistry, everybody's favourite topic or subtopic in biochemistry. That's none of my bismuth. Hilarious Kermit. Enantiomers, mirror reflections, non-superimposable, dash, wedge, 2 lines, dash, wedge, 2 lines. You can see it on the paper. You can see it here. Obviously, that's the mirror reflection. Is that what students always write in exams? No. Practice it. Amino acids. Okay, they're all optically active. Why not glycine? Because it's got 2 groups the same. So cannot that. They're optically active, rotate the plane of polarized light in equal and opposite directions. Rosa's red Toblerone is a prism. I'm not moving on. The Corn rule, CORN. C is the carboxylic acid group with hydrogen at the back. R is the alkyl group, and N is the amino group. This is a good 2-hour lesson, so I'm flying through this one here. CORN, put the carboxylic acid at the top, and then orientate the amino group and the alkyl group with the hydrogen at the back. If you've got a 3-dimensional brain, congratulations, you're going to smash this part of the syllabus. If not, you better start practicing now. Ha ha ha. The proteins are composed of L enantiomers. 19 out of 20 exist as L. Glycine is not optically active. It does not have a chiral carbon. It has 2 hydrogens, the same. Carbohydrates, okay, all simple sugars are chiral molecules. We've got 2 enantiomers of glucose, D and L. D refers to the OH group on the terminal carbon, which is highlighted with a red star here on the right, and L is on the left. Okay, glyceraldehyde. It's dextrorotatory, and L is levorotatory, and that refers to the plane of polarized light. How do we assign bigger monosaccharides? Well, it gets more complicated pretty quickly, and you would need to look at the position of the OH group on the first carbon from the CH2OH at the bottom in a Fischer projection. Filthy, tricksy hobbits. Diastereomers, diastereoisomers, optical isomers, more than one stereocenter. It's going to have 3, 4, 5 different stereocenters to give you diastereomers. If it's only different only one chiral carbon atom, they are epimers. Epimers, mannose, glucose, galactose. Okay, go through those. Identify DDLD, very good. Enantiomers, Haworth projections, Haworth projections, nobody cares, tomato, tomato. In aqueous solutions, glucose forms a ring, so dissolve it in water, it forms a ring structure. Restricted rotation, 2 isomers are formed, alpha and beta. Look at alpha on the left with the OH group, which is beneath the plane of the molecule, and the beta glucose to the north of the molecule. We go to cyclic monosaccharides. D plus glucose can isomerize into beta D plus glucose. What's the difference? Again, it's beneath the plane of the molecule is the hydroxyl group in the alpha D plus glucose, and it's above the plane of the molecule in the beta D plus glucose. It's all about the orientation of the hydroxyl group. The same applies for fructose. Practice those. They could be on the exam this Friday. Fructose is there. Pause it, learn it, move on with your lives. Lipids, these are fats and oils with carbon-to-carbon double bonds. Double bond is a sigma bond and a pi bond. We're going to have cis and trans or E and Z isomers. Okay. Oleic acid is cis, can be cis, and allydic acid is trans. Trans opposite, cis same side. Remember, E is trans, E is enemies, and Z is cis. Transoleic and cis-oleic. Why is it cis when it's got a kink in the chain? Because it's cis around the double bond, not cis around the orientation of you observing the molecule. Replay that 3 times, you'll remember it. Cis to trans hydrogenation using a nickel catalyst. This is partial hydrogenation, double bonds to single bonds in the lipid or the fat. Advantages of hydrogenation: the liquid becomes semi-solid so you can make margarine, decreases the rate of oxidation, and controls the feel and plasticity of the oil. Disadvantages: they're not very good for you. It increases the amount of LDL or bad cholesterol, and they're a low quality energy source. You get them all from McDonald's and Four Fingers. Here is starch, carbohydrate, which we've got in plants, which we all eat. We can metabolize starch because it's made of a polymer of glucose molecules with a condensation reaction bonding each of the rings together. We've got amylose and amylopectin with 1,4-glycosidic bonds and 1,6-glycosidic bonds. Amylose, by the way, which is here, it's going to have a 1,6 linkage as opposed to 1,4 and 1,6 in amylopectin. Glycogen is a polymer of alpha glucose with 1,4 and 1,6 glycosidic links. But it gets quite tricky because you've got beta glucose and beta 1,4 glycosidic links. Look at the bottom, study it, learn it, move on with your lives. Cellulose, we can't digest cellulose. It is the building blocks of plants. There's a man made of plants, which clearly is fictional and not true. But we need them for dietary fibre, for the passing of the waste of our food through the usual method. Dietary fibre, yes, there we go. Vitamins, stereochemistry. Vitamin A is involved in vision, is cis and transretinal, which is involved with rhodopsin in the eyesight of human beings. And you need to talk about the rods and the cones, and I just mentioned the role of rhodopsin, which was a question 2 years ago, which is a large conjugated protein molecule. Here is cisretinal with the opsin to make a rhodopsin. What have we done? We've got a C double bond N. We have made a secondary amine, which is the sort of thing that they like to ask. How does this reaction happen? It's photolytic, so light induces the reaction. No surprises there, it's how you see. Without rhodopsin, you would not be able to see. Cis versus trans. Thank you very much indeed. Have a great day. Good luck for Friday.